Hello, I'm Annemieke Aartsma-Rus, and in this presentation, I will give some more information on when to use which animal model. And there are, of course, a lot of different animal models possible. Most people use mice, but for many diseases, there's dog models, there's fish models, there's fruit flies, there's worms, there's many others. And of course, you should not use an animal model if you don't have to. If cells can give you the answer to your question, use cell models, and especially now with induced pluripotent cells, there is a lot more opportunity here. However, there are questions where you need an animal model to answer, and only then should you use them. And then the question is, which one should you use? Well, why are people mainly using mice? Because they're easy to use. So they're easy to breed, they get really large litters, and the gestation time is 3 weeks, so that's relatively quick. And also they mature quickly. So at 8 weeks, the mice is an adult and can produce offspring. They're small, so that means that the space is limited and the costs are limited, but also that if you want to teach the treatment for the first time, you need small amounts because a mouse weighs 25, 30 grams. So that is a lot less than if you, for instance, would use a rat that weighs 10 times to 20 times more. That means that you would need 10 to 20 times more of your compound, which can make things a lot more expensive. And for a first test, you want to keep things as cheap and easy as possible. But what about flies and worms and fish? Because they're even easier to breed. It's really easy to generate knockouts. They're small, so the amount of compound that you need is very, very little. There's functional assays, how these animals swim. So again, I'm from the mussel field. So there's tests on how quick these animals can swim, how quick the Drosophila can fly. The gait in the C. elegans worm has been studied, and if animals have problems with muscle disease-related proteins, you can see that there's problems with the gait and the speed at which the worms crawl. So you can use these things, but you have to bear in mind that, of course, we are much further from the fish, the fly, and the worm than we are from other mammals. So they're useful for first screen, but then you have to validate in mice, which are closer to the humans. And especially genetically, there's often a lot of difference between the worm, the fish, and the fly, and the mammals. Then for larger animal models, these are closer to the human situation and closer to the human anatomy and physiology. So dogs and pigs are now also often an option. And often these models have a more severe pathology than the mice. However, it's extremely expensive to keep them. There's more heterogeneity between the different animals. But then again, because generally litters are smaller, it's very expensive to keep them. You can only use very small groups. So there is more variability. So ideally, you would need larger groups to get a consistent, robust answer to your question. But now you can use only smaller groups because of costs and availability. And of course, especially for dogs, there's more ethical concerns. However, sometimes it is the only option when the only model that is available is a large animal model, for instance, because the disease is not recapitulated in a mouse, or because there is a dog model available or a pig model available. Well, it seems to be impossible to make a mouse model. And also sometimes you need the larger animal model because scale is an issue. And an example here is biodistribution for viral vectors. It's really easy to body-wide treat a 25-gram mouse. A young human patient will be 1,000 times heavier, probably weighing around 25 kilos. That means that the scale is an issue, and you can't really predict from what happens in a mouse, what will happen in a human. So you need to see, can I have the biodistribution that I need in a larger model? And then the only model available is the dog model or a pig model or another large animal model. Then there's also some consideration because there's now a lot of gene or transcript targeting therapies, like genome editing targets the DNA and antisense oligonucleotides target the RNA transcripts. And these are gene or transcript specific. So here there is a problem that the human gene often has mismatches with the mouse gene or the fish gene, etc. So you really need your human-specific genome editing guide RNAs or antisense oligonucleotides. And therefore, the human cell cultures like the iPSCs may be a better option for proof of concept and off-target analysis. There's no point in looking in off-targets for RNA or genome editing therapies in the mouse because the potential off-target in the mouse will differ from the off-target effects in the human. And then if you want to study delivery, biodistribution, etc., again, ideally you have in vivo models, but then the best option is the humanized model so that you can use the compound that you will use in clinical trials, be them guide RNAs or antisense oligonucleotides. So the one that you will test in trials, it's ideal if you can test that in vivo, in animal models, and then humanized mouse models that have part of the human gene or the whole human gene with a mutation in them are ideal. But again, you cannot use these models to study off-target effects because the rest of the genome will still be of mouse origin and not human origin. So thank you very much. If you have any questions to me, feel free to email me. And I hope you found this presentation useful.